Today I'm doing a Cambridge IGCSE past paper talk through of Biology Paper 4 Theory Extended from June 22. Some students were studying the activity of yeast. They made a fact file as shown. State the kingdom in which yeast is classified. It is fungi. State the process that occurs in mitochondria to provide energy for yeast cells. So the release of energy occurs by aerobic respiration. That's respiration involving oxygen use. Yeast cells make the enzyme sucrase. Sucrase catalyzes the breakdown of sucrose to glucose and fructose. Enzymes are made of protein. Explain how the shape of a sucrase molecule is related to its function. So sucrase is the enzyme, and the enzyme sucrase has an active site. And the key thing about that active site is that it is complementary to that of the substrate molecule, which in this case is sucrose. The students made an extract of sucrase from yeast cells. They investigated the activity of the sucrase extract at different pH values. They determined the rate of reaction at each pH. They then calculated the rate of each reaction as a percentage of the fastest reaction to give them the percentage activity of sucrase. The results are shown. Describe and explain the effect of pH on the activity of sucrase shown in figure 1.2. That's worth 6 marks. So describing is saying what you see, and explaining is saying why. So I've started by saying that as the pH increases from 2 to 6, there is an increase in sucrase activity. Let's talk about what's going on. We're going to say, because this is the optimum pH, we're going to see the most number of enzyme substrate complexes are formed here. And then above pH 6, we can see a decrease in enzyme activity. So from pH 7 to 12, there is a decrease in enzyme activity. Let's say why. Because the sucrase enzyme is denatured, meaning the substrate molecules no longer fit to the active site. Mammals have a double circulation. That means that the blood flows twice into the heart for every once it passes around the body. Figure 2.1 is a diagram of a section through the heart. The arrows show the direction of blood. State the name of the chamber of the heart with the thickest wall. So we're looking at this chamber here. Remember, the two sides of the heart are flipped. So that means it's the left side. It is the left ventricle. D is a vein. State the name of this vein and describe its structure. So we're looking at this vein here. So blood is coming from the body, it is deoxygenated, and it's going to enter the heart via the right atrium up here. Its name is the main vein. It's the vena cava. What about its structure? Well, because it's a vein, it has a wide lumen. It has thin walls made up of muscle and elastic fibers. And then because that blood's flowing at such low pressure due to that wide lumen, we're going to have valves which stop the backflow of blood. You don't really need that roll at the bottom. I'm just bulking out my answer a bit. Identify the structure labelled X and state its role in the heart. So look for X. These valves here, the valves are in the pulmonary artery. Remember, the aorta and the pulmonary artery contain semilunar valves. What is the purpose of that? To stop backflow of blood into the right ventricle. And effectively make sure that blood keeps flowing in one direction. Figure 2.2 is a diagram that shows the double circulation of a mammal. The arrows indicate the movement of oxygen and carbon dioxide in and out of the blood. So we'd expect oxygen to enter and carbon dioxide to leave at the lungs. Here are the capillaries in the respiring tissues. This time you'd expect carbon dioxide to enter the blood and oxygen to leave because those cells need to respire. Shade the blood vessel in figure 2.2 that transports blood with the highest oxygen concentration. So the highest oxygen concentration will be found in the blood of the pulmonary vein because remember that's coming from the lungs. It's going to enter the heart at the left atrium. Describe the evidence shown in figure 2.2 that the mammal has a double circulatory system. First of all, there's that separation of the left and right sides of the heart, meaning that oxygenated and deoxygenated blood don't mix. Then we could define it just saying that what I said about the double circulatory system, which is that blood enters the heart twice for every one complete circuit around the body. Explain the advantage of the double circulatory system. Well, remember I've already said there's the separation of the left and right side, meaning that you have a separation of the oxygenated and deoxygenated blood. And what that means is that you only have oxygenated blood going to the respiring cells. The last thing you want is carbon dioxide heading there because it's not going to do anything. Equally, you don't want oxygenated blood returning to the lungs for oxygenation because it's silly because there's already oxygen in it. So the main thing here is there's a more efficient supply of oxygen, and what comes with that is a more efficient supply of nutrients such as glucose and amino acids to body cells. It also has the knock-on effect of allowing more time for gas exchange, and it prevents damage to the lungs. The pancreas is an organ that has roles in the digestive and hormonal system of humans. 3.1 shows part of the alimentary canal and some of the associated organs. State the names of organs J, K, and L. So this large organ at the top is the liver. And then K is where bile is stored. It is the gallbladder. And then under the stomach, you have the small intestine or duodenum. The pancreas secretes hormones into the blood and enzymes into the pancreatic duct. The enzymes are released into the alimentary canal. Complete table 3.1 by stating the hormones and enzymes that are secreted by the pancreas. So enzymes, remember, end in A's. So we're talking amylase, protease, and lipase. Use the number of lines to guide you. In terms of hormones, we're talking about control of blood glucose levels. So really we need to talk about insulin here, which lowers blood glucose, and glucagon, which increases blood glucose levels. Chloride ions also move along the pancreatic duct. CFTR proteins in cells lining the pancreatic duct move chloride ions out of the cells into the duct. Figure 3.2 is a diagram of a cell from the lining of the pancreatic duct showing the location activity of CFTR proteins. These are chloride ions. Look, they're moving from an area of low concentration to high concentration. I feel like this question may be about active transport. Explain how CFTR proteins move chloride ions across the membrane. Perfect. CFTR proteins use active transport to move chloride ions against, that's key, the concentration gradient, or you could say from an area of low concentration to an area of high concentration. Remember, because it's going against the concentration gradient, it's going to require energy from ATP. The movement of chloride ions into the pancreatic duct causes water to move from the cells into the duct to help the flow of liquid in the duct. Explain how water moves from the cell shown in figure 3.2 into the pancreatic duct. It's going to use osmosis, remember, because what will happen is the fact that you have more chloride ions here, it's going to mean that there's a lower water potential in the pancreatic duct. And remember, water moves from an area of high water potential to an area of low water potential. So the water is going to move in this direction by osmosis. And remember, your definition of osmosis includes the presence of a semi-permeable membrane. So chloride ions in the pancreatic duct lower the water potential. That means water potential. Obviously, feel free to write it out in full. Water moves from inside the cell into the pancreatic duct. By osmosis across a partially permeable membrane. If CFTR proteins do not move chloride ions, the liquid in the pancreatic duct becomes very sticky and the duct becomes blocked. Blocked pancreatic ducts are one effect of cystic fibrosis, which is an inherited disease. Cystic fibrosis is used by mutation of the gene that codes for the CFTR protein. Figure 3.3 shows the pedigree diagram of a family that have 2 people with cystic fibrosis, person 5, person 7. The allele that causes cystic fibrosis is a recessive allele. Describe and explain the evidence shown in figure 3.3 that cystic fibrosis is caused by a recessive allele. First of all, we're going to point out what I've already mentioned, that person 5 and 7 have cystic fibrosis, whereas their parents 2 and 3 don't have cystic fibrosis, so by definition they must be carriers, which is indicative of a recessive allele causing the disease. So 5 and 7 have cystic fibrosis, whereas their parents do not. Their parents must therefore be carriers of this recessive allele. Person 7 is expecting a child with a man who's heterozygous for cystic fibrosis. Complete the genetic diagram to predict the probability of person 7 and the man having a child with cystic fibrosis. Use these letters for the alleles. So person 7, okay, that's a female with cystic fibrosis. We know that it's a recessive disease, so by definition, her genotype must be lowercase a, lowercase a. The man is heterozygous, meaning that he has 2 different alleles. Therefore, he must be uppercase A, lowercase a. So, parental genotypes. Let's go. Man, woman. Already worked that out. So his sperm are either capital A or lowercase a. Her eggs are all lowercase a. Let's draw the Punnett square. Doesn't matter what order you do this. That's the woman. That's the man. So, offspring 1. Offspring 2, 3 and 4. So here are the genotypes of the offspring. The phenotypes are going to be healthy, cystic fibrosis, so it's a 50% chance, or 1 in 2. Researchers investigated the effect of adding cattle manure, cattle feces, to fields where snap bean plants were grown. Cattle manure contains protein. Explain how protein in the cattle manure is converted to the type of ions that plants can absorb. So we need to get that protein broken down into firstly amino acids. Then we need to deaminate those amino acids to make ammonium ions. Then they go through nitrites and then finally into nitrates, which can be absorbed by active transport by the plant roots. So protein in cattle manure. Name the enzyme that does this is broken down by proteases into amino acids. Deamination takes place, which produces ammonium ions. Name the type of bacteria in the soil. It's nitrifying bacteria. Convert those ammonium ions into nitrites and then finally nitrates. You need quite an in-depth knowledge here of nitrification and protease use. Snap bean plants are legumes which have root nodules that contain nitrogen-fixing bacteria. Figure 4.1 shows some root nodules. Suggests the advantage to farmers of having snap bean plants that have a large number of root nodules. So let's say what those nitrogen-fixing bacteria do. So nitrogen-fixing bacteria add nitrates directly to the soil, meaning the farmer needs to apply less manure or fertilizer. The researchers investigated the effect of adding cattle manure to fields of snap bean plants. Field 1 was treated with a small quantity of cattle manure. Field 2 was treated with a medium quantity. Field 3 with a large quantity, and then here's our control with no cattle manure. The researchers counted the number of root nodules on samples of plants from each field. Calculate the percentage increase in the average number of root nodules per plant when snap bean plants were grown with a large quantity of cattle manure, field 3, compared with no cattle manure, field 4. Give your answer to 2 sig fig. So we need to do change over original times by 100. So what's this difference here? This is 96. This is 156. So the difference here is 60. So we do 60 over 96 multiplied by 100. 62.5% to 2 significant figures is 63%. When large quantities of manure are put on fields, it can lead to eutrophication of streams and rivers. This can lead to the death of fish. Describe how eutrophication of streams and rivers can lead to the death of fish. So what happens is that adding manure to fields leads to the leaching or washing of nitrate ions into rivers. What sort of plants are in the rivers that will really benefit from that? It's the algae. This leads to algal bloom. So that's a huge increase in algae numbers. So although those algae have grown in huge numbers, they soon start dying off due to competition for light. So due to competition for light, algae die. And then the death of the algae provides an awful lot of food for bacteria. So there's an increase in bacterial numbers as these feed on algae. And then because they're living organisms, the bacteria uses up the oxygen in aerobic respiration. Meaning that the water becomes anoxic or lacking in oxygen. So anoxic water means death of fish. The Mulanje cedar is the national tree of Malawi. This species of tree grows naturally only on Mount Mulanje in Malawi. Many of the trees have been overharvested or destroyed by wildfires resulting in deforestation. State the genus name of the Mulanje cedar tree. So zoom in here. First of all, you have the genus name, followed by the species name. So just copy out the genus name. It is Widringtonia. Explain the undesirable effects of deforestation on habitats that are on mountains such as Mount Mulanje. You can kind of take this answer wherever you want. Let's start by saying that deforestation means there are fewer tree roots to hold down topsoil. Without that topsoil being held down, you're going to end up with leaching of minerals into surrounding streams, making that soil less fertile and more prone to soil erosion. Ignoring tree roots, what about the loss of trees themselves? Loss of trees means fewer habitats for animals, leading to a loss of biodiversity. Scientists in Malawi are working to prevent the extinction of the cedar tree in its natural habitat. Explain the benefits to other organisms on Mount Mulanje of conserving the Mulanje cedar tree in its natural habitat. So what would be beneficial about the cedar tree? Well, it provides a habitat for birds so they can nest. It also provides them with shelter and protection from predators. You could talk about the fact that any leaf litter produced by the cedar tree is used by decomposers or provides food for fungi such as mushrooms. 2 marks. I'm going to say cedar trees provide nesting sites and protection from predators. For birds. The seeds of many endangered tree species are kept in seed banks, suggests why it's important to collect seeds from many individual trees of each species rather than just one tree. That's all to do with genetic variation. So lots of trees, and that's important from a sexual reproduction point of view because it means that those genes will be more varied and therefore there'll be some individual trees which are better adapted should the environment change. So this means plants grown from these seeds are less susceptible to disease. Figure 6.1 is a flow diagram showing the events that occur to form a human fetus. So organ P produces a diploid cell and then a haploid cell, which is a sperm cell. So I imagine that organ P is testes. The ovary produces a diploid cell followed by the egg cell. Oh wow, look. Remember, meiosis halves the chromosome number, so I imagine that's process Q. Process R will be fertilization. When those two haploid cells meet, the diploid cell will be a zygote, which is the first cell formed after the egg and cell meet. Process T produces an embryo. That will be mitosis because that's a cell division which keeps doubling the number. Process U produces a fetus. That's when the embryo implants in the uterus, so that's going to be implantation. Anyway, I'm getting distracted. What's the actual question? Complete table 6.1 using information in the flow diagram to identify the cell, organ, and processes. So, organ P, testes. Cell S, zygote. Process Q produces haploid sperm and eggs. I've already said it's meiosis. Process R, which produces diploid cell S. Process R, I said, is fertilization, which produces cell S, the zygote. Process T occurs so that cell S can grow into an embryo. That's mitosis. Process U occurs so the embryo can gain oxygen and nutrients from the mother's blood. That is implantation. State why it is important that sperm and egg cells are haploid and not diploid. Well, you'd keep doubling up the chromosome numbers, wouldn't you? If when they met, you had two diploid cells meeting, you'd go from having 23 plus 23 producing 46 to 46 plus 46 making 92. So the diploid number does not double at fertilization. You could even say what I'm talking about here, e.g. 46 plus 46 making 92. State the function of the jelly coat that surrounds the egg cells. It stops more than one sperm cell fertilizing the egg. Complete the sentences with the appropriate words. The placenta provides a large surface area for the something of oxygen and CO2 between maternal and fetal blood. So how does that oxygen and carbon dioxide move? By diffusion from an area of high concentration to low concentration. Dissolved nutrients also pass across the placenta. Examples of dissolved nutrients are, so what have we eaten and broken down using proteases? Amino acids, glucose that comes from starch, fatty acids, glycerol. Antibodies pass from the maternal blood, giving natural, passive, because you're not being infected with the pathogen, immunity to the baby for some infections that the mother has had or been vaccinated against. Each different type of vaccine contains one or more antigens. Remember, that's the protein on the outside of the pathogen, which is recognized by the lymphocytes, the white blood cells, taken from the pathogen. That's the microorganism that causes the disease, as it says here.